Welcome to Life in Biology. I'm Dr. Joel Graff, and this video is going to be about how a cell can respond to a virus infection. As you can imagine, there's lots of different viruses in this world, and so a cell needs to be able to pick up on the fact that it's getting infected very rapidly, and then it would also want to control a fairly large number of genes so that it could provide some sort of a defense against all the possible virus mechanisms of taking control of the cell, so to speak. So the first thing you have to do if you get infected with a virus is to know that you're infected. And so the viruses are coming. I'm not a very good drawer. Here's an example of a virus that I usually draw in my class. It's just a hexagon with some sort of genome segment in the middle. Once you have attachment and entry and encoding, so I'm kind of combining all three steps there with that little arrow, the virus genome becomes apparent to the proteins within the cytoplasm. We just had a video talking about DNA replication, and when you split apart those two strands of the double-stranded DNA, a helicase did that job. Well, a helicase domain, which I've drawn as a triangle, just like in the previous video, is going to be the component of this protein right here that is able to detect viral genomic DNA or RNA, or any combination of. And then I have, in addition to having just a helicase domain, there's also what I've drawn a K with a circle around it, and that's supposed to represent a kinase. And so the kinase is a protein that will cause phosphorylation of another protein. So we have protein 1 here, and it gets phosphorylated, and it could be a kinase as well. And if it is, then it would go find protein 2 and put a phosphate group on the second protein. And then this way, each step of the signaling there can be amplified, because this first kinase can go and find multiple examples of protein 1 and put phosphate groups on it. Each of those protein 1s could go and find multiple protein 2s to put phosphate on it. So it helps amplify and quicken the response. At some point, the pathway could split into multiple pathways. And in this example, I have 3 different transcription factors being activated. Transcription factors are the proteins that can either induce or repress gene expression. These transcription factors, NF-kappa B, IRF3 or IRF7, and AP1 transcription factors, these are examples of transcription factors that normally hang out in the cytoplasm of a cell. And when they get activated, maybe when protein 2 is in the phosphorylated form, it's able to branch out and activate these other transcription factors. There's more to it than that, but that's a simplified example. Then these 3 different types of transcription factors would end up going into the nucleus of the gene, the cell, excuse me. And in the nucleus is the genome of the cell. And so each of these transcription factors, there might be multiple versions of each one going into the center. They're going to go and they're going to try to find the promoters for which they were designed to recognize. So each one of these transcription factors has a specific DNA sequence, a binding consensus site that it wants to bind to. So each of those can go to the nucleus and turn on a whole set of genes. And it's probably, NF-kappa B can be like maybe up to 500 different genes that it can control. IRF3, there's quite a few. AP1, there's definitely a lot of genes. And so with these 3 transcription factors activated, you get an immediate response of a lot of genes. But in an antiviral response, something that really adds to the increase in antiviral genes is when you get a cytokine. That's a protein that a cell would secrete to communicate with another cell. When you get cytokines being made, they can amplify the message. In this case, with an antiviral response, we're going to focus in on a protein called interferon beta, and then this is the enhantiosome. So the gene itself is just called IFNB. There is the transcription start site. We're getting used to seeing those sorts of things. But if you have the AP1 transcription factors the IRF3 transcription factor, or 7, and the NF-kappa B transcription factors, if all three of those get activated, which is what we saw in the previous slide, those three will end up binding to consensus sites that are in the promoter of the interferon beta gene. And they sit in very tight positioning next to each other, and between the three of them coordinate the induction. The induction means upregulation, increased expression of interferon beta. So you have transcription, which gives you an interferon beta mRNA, and then when it's transcribed, you get the interferon beta protein. And the protein is a cytokine, as I mentioned earlier, and cytokines are proteins that are typically either sent out to the surface of the cell as a membrane-bound protein, or they can be released as a secreted protein. In the case of interferon beta, like most other cytokines I can think of off the top of my head, they are secreted. So, what's the good of realizing that you're infected with a virus? I'm talking from a cell's point of view, and then just sending the cytokines out into the periphery. Well, here is an example where we have two cells. Over here we've got an infected cell, and here we have a neighboring cell. So within a tissue, cells are going to be surrounded by lots of other cells. This cell is infected. This cell can't practice social distancing, and so it's very much in danger of getting infected once this cell passes the virus on. So what you want to do is when that interferon beta gets secreted, you get lots of these. So these circles are cells and these circles are proteins. So these interferon beta proteins are secreted into the extracellular matrix, and they can go and they can bind to a receptor of the infected cell itself. So if it binds to the cell that made the interferon beta, this is called autocrine signaling, self-signaling. And the pathway I'm minimizing here, or I'm simplifying to 2 arrows, but there's a fair amount to talk about with that pathway. And then a transcription factor called ISGF3, as the transcription factor complex goes to the nucleus of that cell and binds to lots of promoters and induces antiviral genes, lots and lots of antiviral genes. So the cell that is infected can fight back against that virus that infected it. Meanwhile, because it did just secrete the interferon beta out into the extracellular space, if the neighboring cell has an interferon receptor, type 1 interferon receptor, interferon beta is considered a type 1 interferon, it will then signal into that cell, and you'll also get antiviral genes being upregulated in the neighboring cell. So between this cell trying to fight off the virus and all of the neighboring cells increasing their antiviral genes, it's quite possible that you can contain a virus infection. So it's possible that many of us get a virus infection and then our antiviral innate immune response kicks in and takes care of the virus before we even know we're infected. If the virus does take hold and start replicating, that's when you might have the adaptive immune response kick in, and we'll talk about that later. But anyway, this just shows that there's lots of genes that get turned on, get upregulated, get induced during a viral infection, both the cell that's infected and all the surrounding cells. We might talk about specific functions of some of those antiviral proteins in another video. Until then, like and subscribe, or not.